Vedantu NEET English. I'm Ms. Gopika, your own biology master teacher. I hope I'm audible and visible to all of you. Please let me know in the chats if I'm audible and visible so that we can start the class very quick. Okay, please let me know if I'm audible and visible. And today we are going to do sexual reproduction and flowering part, the final part, the last part, where we will be talking about the post-fertilization events and about double fertilization, which we could not complete in the last class. Okay, yes. Come on, please let me know if I'm audible and visible to all of you. Let me also just refresh the chats. Okay. Yes, and quickly share the link with all your friends, okay? So all of them joins. Yeah. I can see your chats now. Everything perfect? Hi. Hello. Okay, so let's start with post-fertilization events, okay? So students, basically this chapter, is divided into three parts: fertilization, pre-fertilization, fertilization and post-fertilization, right? So we have completed for pre-fertilization, fertilization, and now we are going to do post-fertilization, which I will be taking the help of NCERT line by line to do that. But the fertilization part, we have to write few notes for that, okay? So I want all of you to quickly share the link with your friends, ask everyone to join, because some of them would have forgotten that 8 o'clock there's a class, okay? Yes, perfect. Okay, chalo. I hope all of you marked your attendance as well, okay? Like every other class you join, you have to mark your attendance. It's very important to mark your attendance. Okay, let's start, students. By then your friends will join. So you know the weightage of this chapter is 5 marks, okay? So last class I have already taught you pollen-pistil interaction, what exactly happens. So I told you the stigma and pollen, there is a chemical dialogue, there is an exchange of chemical dialogue where there is basically a chemical released. The stigma has to accept the pollen grain, the right pollen and the compatible pollen has to fall in the stigma, then only it will allow the pollen tube germination to happen. If the wrong type and an incompatible one falls on the stigma is not going to allow the pollen to grow, right? So for that they have an exchange, there is a chemical dialogue that is exchanging between them, because of which there is going to be an acceptance. After an acceptance, the pollen tube germination continues, where the pollen tube in a 2-cell stage, there is one generative cell, one vegetative cell, and in the 3-celled stage it is going to be 2 generative cell and 1 vegetative cell, which is going to come through the tube nucleus, and there is a tube through the pollen tube, and there is a tube nucleus which actually guides this fellow, okay, which will actually guide this fellow. Clear? Okay, now look here carefully. So what did I tell you? In a 2-celled stage, okay, all these notes will be available on Vedantu NEET English Telegram channel. Okay, 2-celled stage, what is happening? The pollen grain has fallen, okay, the pollen grain has fallen. Now students, this pollen grain will have, one second, this pollen grain will have vegetative cell and it will have the generative cell. Can you see the red inside the yellow? Little light, no? Okay, I'll draw it with a, I think this should be seen. No, it's worse than that. Okay. Yeah, this is bright enough. So this is a pollen grain of a 2-cell stage. That means you are able to see that there is a vegetative cell, right? There is a vegetative cell, and here you are able to see that there is a generative cell, okay, there is a generative cell. Now this will fall on the stigma, and from here what is going to happen, students? The pollen tube growth will happen, right? The pollen tube growth will happen, which will have one male gamete, okay, which will have one male gamete. Okay, this is the one male gamete. Now tell me, what is the fate of this one male gamete? What will be the fate of this one male gamete? The fate of this one male gamete is going to be that it is going to reach the ovary, enter into the embryo sac and go and fuse with the egg, correct? Go and fuse with the egg. I feel it is too bright. One second, students, let me just decrease the brightness. Okay, now it should be better. Yes, tell me, all of you. Okay, I can't see your chats, I don't know why. I think I can see it in the phone better. Pradeep Kumar, I am good, how are you? Okay. So, here what is happening, students, that one male gamete, okay, one male gamete is going to fuse with the egg. This is what is going to happen here, right? This is what is going to happen here. Now, look here, what is the situation here? What about a 3-celled stage, okay? What about a 3-celled stage? 3-celled stage, students, initially this is what happens, right? Initially what is going to happen? There is an entire, vegetative cell, right? There is an entire vegetative cell, and inside the vegetative cell there is going to be one generative cell. This is the scenario here. But after some time, okay, once the pollen tube germination happens, after some time we notice that the generative cell will undergo one more mitosis, okay. The GC will undergo, okay, one more mitosis, and this will lead to what? This will lead to 2 male gamete, okay. This will lead to 2 male gamete plus 1 VC, okay, plus 1 VC. I hope this is clear to all of you. Clear? Fine? Okay, got it. Okay, I hope all of you are marking your attendance as well, okay, marking your attendance as well. Okay, yes, this is done. There are a few reflections, that's why. Okay, now look here, students, what is exactly happening here? You notice that the pollen tube. So, I want all of you to understand this very simple thing, that your pollen tube, okay, when we talk about your pollen-pistil interaction, it is very important. Okay, when we talk about your pollen tube, how it enters, remember the pollen tube germination is happening, okay. First, where is it going to enter? It is going to enter the ovary. From ovary, where is it going to enter? Into the ovule. After the ovule, where is it going to enter? To the embryo sac. In the embryo sac, where will it enter? It will enter into the cytoplasm of the synergids with the help of filiform apparatus. After that, it is going to go and fuse with the egg. So, that is the kind of pathway that you have to remember, okay. First, it will enter the ovary, okay. First, actually, when you look at it, this is the stigma, okay, this is the stigma, style and ovary, right. So, if you notice exactly, the pollen tube is going to start from here, it is going to enter into the style, okay. First is going to be style, then to the ovary, understood. So, if I have to start from the beginning, it will be, I will write the style, okay, then to ovary, then inside the ovule, then inside the ovule where? To the embryo sac, okay. Inside the embryo sac, first it will enter the cytoplasm of the synergids with the help of filiform apparatus, okay, synergids, correct, yes, very good, okay, yes. So, cytoplasm of the synergids, that is your filiform apparatus, okay, and then it is going to fuse with the egg, okay. This is the pathway the pollen tube takes. Clear to all of you, yes or no? Please let me know if it is clear. Quickly let me know in the chat box if it is clear. Okay, done, done, okay, done. So, this is the one, okay. Now, students, double fertilization, which is one of the most important topics, okay, important topics is going to be double fertilization. Here, what we have to learn is that what we just now understood with the pollen-pistil interaction, same thing, okay, we have to understand here, okay, these are the two things, okay. We have male gamete, male gamete, okay, male gamete. Now, we will draw here the egg, okay, we will draw here the egg and we will draw here the, okay, I will change the color here, polar nuclei, okay, we have polar nuclei. I hope the color difference is clear, okay, nice. Now, look here, this is the male gamete, this is the male gamete, okay. this is the egg, okay, this is the polar nucleus. This is the polar nucleus. Now, what exactly is happening here, students? It is simple. Two male gametes, okay, in a 3-cell stage, two male gametes will be there. One male gamete will go and fuse with the egg and it is going to form your zygote, correct. One more will fuse with the polar nucleus to form the primary endosperm cell. So, that is what we learn under triple fusion, okay, that is what we learn under triple fusion, plus and plus, right. Fusion is happening. Then here what is going to happen? First, let us label this, okay, let us label this male gamete, okay, male gamete plus what is this going to be, students? This is going to be the egg, correct, this is going to be the egg. Here also what is this going to be? Male gamete, okay, and this is going to be the polar nuclei, okay, polar nuclei, correct. These two are going to be the polar nucleus, correct or not? Done, okay. Now, this normally the male gamete and egg would have led to the formation of what? Zygote, right. It would have led to the formation of zygote, which is a diploid, okay, which is a diploid figure. Now, this process, male gamete plus the egg to form the zygote, this process is called as syngamy. What is this called as? Syngamy. Clear to all of you? Very, very, very important. Syngamy. Syngamy, okay. So, no, this is not the right place to ask such questions. Whenever I take some other sessions or whenever I ask any comments, you can put it in the comments, okay. I do not want to disturb the flow of the class. Clear? It is a long question to answer, okay. Clear? Now take a drop or go with another course. It's your wish. See, I am no one to tell you to take a drop, okay. I am not a person who promotes drop culture. It is up to you. If you are ready for 1 year whole struggle, you should be taking a drop. Otherwise, you can go for another course. Clear? Okay. So, students, zygote and syngamy. Clear? Now, you look here, male gamete plus the polar nuclei which was present inside the central cell, they are also going to fuse and this is going to lead to, we call something called as triple fusion. What is this called as? Triple, okay, so 1 second. First of all, what they will form? They will form primary endosperm cell. They will form what? Primary endosperm cell, which we call it as PEC. We call it as PEC, okay. So, I will just draw the structure to make you understand this a little better. So, this is the structure, okay. Inside this, student, there is going to be a nucleus, okay. This is what we got, okay. This is what we got. Now, look here all of you. This one, the pink one that I have drawn, if you want me to color, no need to color, this pink border that I have drawn, this is your PEC, primary endosperm cell. This is your primary endosperm cell, correct. Now, this one is going to be a primary endosperm nucleus, primary endosperm nucleus, got it. This will undergo maturation and they will ultimately form endosperm. They will ultimately form what? Endosperm. Now, what is the most important thing here? This PEC is going to be a triploid figure. This is going to be 3n. Why this is going to be 3n? Because here you notice this and this is going to be 2n, right, n and n, and this is 1n. So, this is going to ultimately form what? 3n, right. So, this process is called as triple fusion, triple fusion. Clear to all of you? Clear to all of you? Very simple. Look here. Normally, a male gamete will fuse with the egg to form zygote, correct. Now, that zygote, that formation is called as syngamy. Now, the other male gamete which we saw in a 3-cell angiosperm only, that male gamete will fuse with the polar nuclei which is present in the central cell to form something called as the primary endosperm nucleus or your primary endosperm cell. Okay, inside that primary endosperm cell, you will see the primary endosperm nucleus. This will mature to form your endosperm. Now, what is this endosperm going to do? This endosperm is going to provide, okay, it is going to provide nourishment it's going to provide nourishment for the growing embryo, we can tell, or zygote you can tell, but embryo is a better word, okay, for the growing embryo. So, that is why endosperm formation will happen first, then the zygote formation will happen. Why? Because if food is not there, now once you are born, let us imagine the entire world there is no food. First you are born, then the food was done. It will be like you are going to starve. First food should be formed, then you should be given birth. Clear? So, that is what here is happening. So, this is together. So, triple fusion plus syngamy together forms your double fertilization. If it is clear, please give me a thumbs up in the chat box, all of you quickly. Please give me a thumbs up. Last notes links are not working. Huh? I will share it with you. Okay? I will share the notes with you. Clear? Hi Shailaja, ready? Okay. So, here what is happening? Endosperm will be formed first. Endosperm provides the nourishment for the growing embryo. So, this is what is happening. Male gamete plus egg, zygote, normal. Male, female, normal gamete formation. Okay. Here what is happening, students? One more male gamete will fuse to the polar nuclei to form your primary endosperm cell. Inside the primary endosperm cell, there is going to be primary endosperm nucleus, which on maturation will form your endosperm. Again, these people are 3N. Right? These people are 3N. Why they are 3N? Because your polar nucleus is 2N. N plus N. Correct. And here you have male gametes 1N. So, it is going to be 3N, and that fusion we call it as triple fusion. Okay. That fusion we call it as triple fusion. Clear? Done? Now, here, so what is double fertilization, students? Double fertilization is basically syngamy plus triple fusion. Syngamy plus triple fusion is together called as double fertilization. Students, in which type of angiosperm this happens? In which type of angiosperm does this happen? Come on, quickly in the chats. Okay, quickly in the chats. Which type of angiosperms? Okay, this happens in a 3-celled, okay, angiosperm. Okay, 3-celled. Why? Because triple fusion wouldn't happen in a 2-cell stage. Only one male gamete is there, means that one male gamete will fuse with the egg and it will form the embryo. There is no tension in that. Only in a 3-cell stage where one more male gamete is formed, that will go and fuse with the with the polar nuclei to form the primary endosperm nucleus. Okay, clear to all of you? Yes or no? Very soon, Nishu. Yes, Jessie, very true. Okay, done. Done. Now, artificial hybridization. Okay, artificial hybridization, one again very important process, students. Normally in a flower, okay, normally in a flower, what will be there? We will have the anther, okay, we will have the anther, and then we will have the stigma style ovary, okay, stigma style ovary. Correct. This is what a flower will have. Let's just give a base to this. Okay, this is a flower. Okay, this is a flower. Now, students, this flower, from this flower, what I am going to do? So, this is a flower which has both anther and stigma. Okay, it has anther and stigma. Correct. Now, this artificial hybridization is a process if plant can select. If plant can select which is the one that is right compatible for them, which is the one which is not compatible for them, then even humans can do that. So, that is where farmers come in. Farmers will be like, you know what, I want to do artificial hybridization, where it will not allow the self pollen to fall on them and at the same time unnecessary pollen to fall on them, but instead they dust the pollen of their desired choice on them. This kind of a process is called artificial hybridization, which happens in 2 steps. First is called as emasculation, second is called as bagging. Okay, bagging. Clear? So here what is going to happen, students? Let's go to the next step. So in the next step, you will notice that here, okay, one second, diagram is always the better way to understand than, you know, just talking. So here you will notice that in this flower, okay, in this flower you notice that the anther has been removed. So this process where, okay, the process where we remove, okay, removal of anther from the flower, okay. So this removal of anther, students, it can happen with the help of forceps, okay, that is any tools to remove that, okay, any tools to remove that. So you can just use any tools to remove the anther. So removal of anther from the flower, this process is called as emasculation. What do you call this process as? Emasculation, okay, emasculation, emasculation, okay. So emasculation is a process by which where you remove the anther from the flower to avoid self-pollination from happening, okay. So the main thing that we are trying to avoid here is what? We are trying to avoid this from happening, okay. So this is what we are trying to avoid, no self-pollination, no self-pollination. Clear? No self-pollination. This is what we are trying to avoid here. Okay. Now, look here, students. See, after this process happened, don't you think that there will be other roadside people also looking at the stigma and be like, oh, I want to dust my pollen also on this. There will be many random pollen grains who are waiting to come and dust on the stigma. So, do you think the farmer will allow for that? No. The farmer will be like, no, I am not going to allow for this. So, I am going to cover this emasculated flower with the help of a butter paper. With what, students? With the help of a butter paper, this emasculated flower is going to be covered. Okay, butter paper, some people even use plastic covers. We are going to cover this paper. This process of covering, okay, covering, or let's say bagging, okay, I will not use the word now, I will just use covering, covering the emasculated flower. Emasculated flower is called as what? It's called as bagging. It's called as bagging. Done? Done, 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 done. Yes, all of you, please give me a thumbs up. Energy is low, I want more energy because I am standing on antibiotics. As antibiotics come down, my energy will come down, okay. Yes, I caught a pretty bad fever, so yeah. So come on, I need all of your energy, okay, all energy should come to me. Done? All of you understood? Understood? Is there anyone who hasn't understood anything in this class, okay, who needs more explanation? So what's happening here, students? Very simple. So the pollen grains, we do not want self-pollination to happen. So we have removed the anther with the help of a forceps. That process is called as emasculation. After doing emasculation, there is chances that roadside pollen grains will fall, some other pollen grains will fall, undecided pollen grains. To avoid that, we do something called as covering the emasculated flower with the you know, butter paper. Okay, I'll write that also. Okay, with butter paper, which is called as bagging. Okay. Now, after bagging, what is going to happen? Listen to me carefully. After bagging, students, we have still our stigma, okay, who is pavam waiting when will a pollen come and fall. Okay, stigma is there. Now, the farmer will decide. Okay, the farmer, the person, whoever is doing this, okay, that person will decide. Thank you so much. Thank you so much. Yeah. So, that person will decide to put a desired pollen grains. Okay, to add or to drop the desired pollen grains from the anther of another flower or anything according to their wish. Okay, it will dust. So, this is your desired pollen grains. Okay, desired pollen grains. So, this desired pollen grains will be dusted on the stigma. Can you see that? Yes, ma'am, we can see, ma'am. Okay. So, like this, desired pollen grains, it will be dusted. Now, after this, students, after this, I cannot, copy paste that option. I don't have. Otherwise, I would have not drawn this many flowers. Okay. So, after that, what will happen? The stigma will be happy. Okay. All sandosham. Okay, like this. It will have the desired or the pollen grains that was supposed to be, added by the person, and after that rebagging will happen. Okay, what will happen? Rebagging will happen. Students, if rebagging is not done, again there is chances that contamination happens. So, here we do something called as rebagging. After dusting the desired pollen, we do rebagging to avoid any kind of contamination. Clear to all of you? So, these are the two main processes that happen in artificial hybridization. Okay, that is emasculation and bagging. Emasculation is where we remove the anther, bagging is where we cover it with a butter paper or a plastic cover to avoid any undesired pollen from falling. Okay, then we dust the desired pollen, and then after dusting the desired pollen, we will do something called as rebagging. Okay, we will do something called as rebagging. So, I hope this step is clear for all of you. Yes? Okay. Now, so here this is what is given in your NCERT. Okay. Can you all see NCERT? So, as you shall learn in chapter on the plant breeding, a breeder is interested in crossing different species and often genera, combining desirable characters to produce commercially superior varieties. Okay, that is why we do artificial hybridization. So, a breeder will select. Okay, artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiment, it is important, like how Mendel did. Okay, it is important to make sure that only the desired pollen grains, okay, only the desired pollen grains are used for pollination, and stigma is protected. See, that is why I told you, after dusting we have to make sure that the stigma is not contaminated. So, stigma is protected from contamination from unwanted pollen. This is achieved by what is the two process? Emasculation and bagging techniques. Clear to all of you? Easy? Yes? Clear? Okay, now moving forward. Okay, moving forward, if the female parents bear bisexual flowers, removal of anthers from the flower bud before the anther dehiscence. So, here we have to be doing female parent bears bisexual flowers, we remove. Okay, so here removal of anthers from the flower bud before the anther dehiscence, usually with a pair of forceps, is called as emasculation. Okay, it is called as emasculation. Now, emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, okay, butter paper, to prevent contamination of the stigma with unwanted pollen. This is called as bagging. Okay, this process is called as bagging. Clear? Now, when the stigma of the bagged flower attains receptivity, that is mature pollen grains are collected from the anthers of the male parent, are dusted onto the stigma, the flowers are then rebagged. See, exact what we drew, okay, and then the flowers are allowed to mature. Clear to all of you? Yes or no? Done, done, done? Now, we are going to learn about post-fertilization events. So, post-fertilization events means what, students? Here, post-fertilization events means we are talking about how the ovule is going to form the seed, okay, how the ovary becomes the fruit, right. This is what we are going to learn in your post-fertilization events, because in fertilization or pre-fertilization events, we studied how we can form the gametes. In fertilization, we learned how to fuse them, and then now in post-fertilization, we are going to see what is the ultimate goal of a plant to do sexual reproduction. Okay, what is the ultimate goal for the plant to do sexual reproduction? So, look here, students, we will learn few from NCERT and we will learn otherwise. So, cells of these tissues are filled with reserve food materials. What are they talking about? They are talking about your primary endosperm cell. okay, primary endosperm cell, they are trying to say that these people will have the reserve food materials, okay. Reserve food materials, why? Because they have to provide everything that they have for the growing embryo, otherwise how will the embryo grow, right. So, whatever food reserve material they have to give everything for the growing embryo, okay, and are used for nutrition of the developing embryo. In most common type of endosperm development, the PEN undergoes successive nuclear division to give rise to free nuclei. This I will show you with the help of a diagram. This stage of endosperm development is called as free nuclear endosperm, okay. Subsequently, cell wall formation occurs and the endosperm becomes cellular, okay, endosperm becomes cellular. Now, the number of free nuclei formed before cellularization varies greatly. Now, students, the coconut water that we drink, the tender coconut water, what is that called? Elnir Vallam in our language. In your language also, I think Elnir only, no? In Kannada also it's Elnir. In Tamil and Telugu, please tell me. The coconut water from tender coconut, okay, from tender coconut, the water that we drink, the coconut water, okay, that you are familiar with is nothing but a free nuclear endosperm, okay, and the white kernel that you eat, that we eat, no, the white thing, the surrounding white kernel is the cellular part. White kernel is the cellular endosperm. This is very, very important. Clear? What do you tell in your language? Tell me in the chats. Faster. So, now students, look here, whatever we just understood, okay, whatever we just heard now, we are going to, with the help of a diagram, we are going to understand, okay. 1, 2, 1 to 2, okay, 2 to 4, okay. Okay, now look here students, what did we study? They told that the cell that you are talking about has, okay, has a nucleus, okay, it has a nucleus and it has all the food reserve, right, it has all the food reserve. Now, what are they talking about? They are talking about your PEN, primary endosperm nucleus, and they are talking about your PEC. Okay, so they will undergo division. Now, when they are telling about they undergo division, what are they mainly talking about? They are mainly talking about this nucleus undergoing the division. So, here they are saying that these people, okay, these people are going to undergo the first karyokinesis, no space, no space. Okay, yes, this is your okay, endosperm, right, like a pre-endosperm types, okay. Kobari Nilu, oh, is it? Elanir only in Tamil also. Which is this language in? Kobari Nilu is Telugu, okay. Yeah. So, now look here students, first scenario what is going to happen, this nucleus has to undergo your karyokinesis. Karyokinesis will happen without cytokinesis, okay. Karyokinesis will happen without cytokinesis, that means from one cell stage to two cell stage, okay, one cell stage to two cell stage. That time also your zygote is going to be here, okay, your zygote is going to be here. Let's just color it like this, okay, zygote is going to be there. Here what is going to happen students? Again same thing is going to happen, karyokinesis will happen. Karyokinesis will happen, okay. Here I will write about cytokinesis together, okay, don't worry about that. Here what is going to happen? 1, 2, 3, 4, okay. 2 to 4 has happened, 2 to 4 has happened. Again here your slowly your zygote is trying to become embryo, okay, your zygote is trying to become embryo here slowly, okay, that is what is happening. Again next stage also same thing is going to happen here also. Okay, here also Karyokinesis is going to happen. Now, here what is going to happen, students? Here 1, 2, 3, 4, 5, 6, 7, 8 is going to happen, right? 8 is going to happen, and here slowly we start seeing the embryo. Okay, we start seeing the embryo. Clear? I'll explain it to you once again, don't worry. Just try to understand. Okay, so what are they telling? They are telling that, okay, pen is primary endosperm nucleus. Okay, NEET. Okay, so look here, students, here what is happening? Your primary, your peck, okay, your peck and your pen is there. Now, these people are going to provide the nourishment for the growing embryo. So, even if I draw a kuti zygote here, okay, like this one zygote here also, it has to continue with, it will start with karyokinesis and we will see two nucleus formation. Karyokinesis is what? Karyokinesis is where the nucleus division happens, okay, division of nucleus. Clear? Those who do not know that, division of nucleus. Okay, so karyokinesis, again karyokinesis happen. Now, please remember here, there is no cytokinesis that is happening. If cytokinesis doesn't happen, students, then obviously wall formation is not happening, right? Cell wall formation, that wall formation is not happening. Here also same thing, no cytokinesis. Okay, no cytokinesis. If cytokinesis is not going to happen, definitely that is going to be a free nuclear stage, right, a free nuclear stage. So, here you notice that this is your zygote, okay, this is your growing zygote, that means again maturity has to come. So, this is your zygote and slowly you will start again zygote formation, by here it has become embryo. Okay. So, this is going to be your free nuclear, what is this going to be? Your free nuclear endosperm, free nuclear endosperm. Clear to all of you? Understood what they meant in NCERT? What they meant in NCERT? Free nuclear endosperm. Okay. Now look here, karyokinesis happen, no cytokinesis happen, and this is your free nuclear endosperm. Here what are the example that they have given you? Here example that they have given you is tender coconut water. Tender coconut water is the example given in your NCERT. Now, students, after the free nuclear endosperm division, okay, after the free nuclear endosperm division, let us say the cytokinesis happened, okay, cytokinesis happened. If cytokinesis happened, then what is going to be the situation? What is going to be the situation? It is going to be, just draw it here only, okay, like this, the same thing there. If I had an option to copy paste, I would have done. Let me just try. I think it will not. Oh, see, this is the problem, okay. So, here same thing, 1, 2, 3, 4, 5, 6, 7, 8, okay, and then you have embryo in between, okay. This is your embryo, correct. Now, cytokinesis has happened means what, students? You will start seeing the cell wall formation like this, okay. Can you see? Like this formation will happen, okay. So, students, what has happened here? What has happened here? Slowly from a free nuclear endosperm, we got a, okay, we got a free nuclear endosperm, we got a cellular endosperm. We got what? A cellular endosperm. So, here what is the example that they have given you for cellular endosperm? Example, they have given the white kernel that you eat from the tender coconut. Clear to all of you? Is this clear? Please let me know in the chats. Please let me know in the chats. You people are not commenting your favorite diagram, nothing you people are doing, okay. Come, study and go, that's all. Okay. Okay. So, here developing embryo. Okay. So, cytokinesis will happen, that means cell wall formation will happen. Clear? Cell wall formation will happen. Done? Okay, cell wall formation. Done? All of you understood this? Okay, this is your cellular and nuclear endosperm, what is mentioned in your NCERT. Now, let me see if I have an empty slide. I want to show you people one more thing. Okay, here I will just show you cytokinesis. Okay, just only cytokinesis. Okay. I saw the comment. Ma'am, speak few words for us. Theriyadhe Tamil. Tamil theriyadhu students. Konjam konjam theriyum. With konjam konjam I cannot talk. Okay. Now come back to class. So cytokinesis. So here what's happening, students? A normal cell. Let's take the what we drew there only. Okay. So if you take a cell like this, if it is in a free nuclear division, okay, if it is in a free nuclear division, after that if cytokinesis happen, there will be cell wall formation. Clear? That will lead to the cellular endosperm. Now the cellular endosperm, 1, 2, 3, 4, 5, 6. Cellular endosperm will do what? Cellular endosperm also will provide the nourishment for the growing embryo. Okay, it is going to provide the nourishment for the growing embryo. Clear? Okay. Now here also same thing. See, cell wall formation. Okay, like the cell wall. Here also cell wall. Here cell wall. Cell wall. Like this formation will continue. Clear? Like this cell wall formation will happen. Now this is very important. Okay. Cell wall formation. This is your developing embryo. Now, few things I want to make you people understand. Here students, endosperm. What do we know about endosperm? We know that endosperm is triploid, 3N. Why? Because it is formed by triple fusion. What is the job of endosperm? To provide nourishment, for developing embryo. So in your NCERT there is a question, why should embryo development precede, why should endosperm development precede embryo development? So what's the answer for that? This one. Because the endosperm provides the nourishment for the developing embryo. Okay. Now students, your zygote has developed or matured or undergone, you know, change, maturation to form your embryo. Okay. Now ultimate goal is what? To form the seed and the fruit. Okay, we are going to come to that. We are going to come to that. Clear to all of you? This is, I hope this is clear for all of you. What is the job of endosperm and what is your, how did your zygote become your embryo? These two things you remember. Okay. Yes. Now here, look here. Embryo develops at the micropylar end. Okay, where does the embryo development, students? Embryo develops at the micropylar end of the embryo sac, where the zygote is situated. Okay, where the zygote is situated. Okay, thank you, Nishu. Okay, now where the zygote is situated? Most zygote divide only after a certain amount of endosperm is formed. Why? Because it needs nutrition. It will be like, please give me some nutrients, right? So this is an adaptation to provide assured nutrition to developing embryo. Okay, what is the need for this, students? To provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, that is embryogeny, okay, important, the embryogeny, where is that formation of embryo, are similar in both monocots and dicots, okay. Now, the zygote give rise to proembryo. So, the zygote has to undergo different subsequent divisions, okay. Octet stage is there, 4 cell stage is there. So, 4 cell stage to 8 cell stage, 8 cell stage to your globular stage to heart shaped stage. All the stages are there, which I will show you in the diagram. So, and then to globular, heart shaped, and ultimately your mature embryo, okay. So, this is what they have shown in the help of a diagram, okay. Now, look here, here see students, what did they say you? Zygote, okay, where this is formed, this embryo, your mature embryo will be formed in the micropylar end. See, locations are important, okay, micropylar end, okay. This zygote will have to undergo division, correct, and it has formed a 2 cell stage. So, we can say like a pro-embryo stage, pro-embryo stage. Now, from this pro-embryo stage, it has formed a globular embryo. See, it has formed a globular embryo. From a globular embryo, it has formed a heart-shaped embryo. Now, remember, everything has a suspensor, okay, everything has a suspensor. Now, from the heart-shaped embryo, it has finally formed a mature embryo, which has a plumule, a radicle, a cotyledon. That means plumule which will end in a shoot and radicle which will end in a root. Okay, I think the diagram is too blur for all of you. 1 second, students. We have solution for every problem. Okay. So, I will not be able to draw the whole thing, okay, because it is going to be difficult for me to draw, but otherwise this is important. So, this is your zygote, okay, this is your zygote. It is going to form a pro-embryo stage, okay, a pro-embryo stage. After the pro-embryo stage, with the help, okay, there is going to be a suspensor, okay. If you notice here, students, you will notice, see how one big head and one tail, like that one region is there, okay. So, here like this one big head, okay, and from the head you have some suspensors that are coming out like this, correct, like this portions, and then you have a globular, okay. We basically draw like this, okay, just draw one globular like this, okay, one globular embryo we got. After the globular embryo, what happened? We got a heart shaped embryo. Again here a suspensor, sorry, a small portion like this, again a suspensor like this, correct. All these are subsequent divisions, okay. It is not like chumma heart shaped is there, so we will draw it. This is all 1 stages, 1 stages this is happening, okay. Ultimately, students, we will form the mature embryo, okay, mature embryo which has a plumule, radicle and a cotyledon, okay. So, we have 1 here and then it formed 2, okay. This is like a pro-embryo I am drawing, okay, and then we students, we have 1, okay, here, and we have suspensor, okay, here, and then the entire thing is covered with stages, right, covered with cells, okay. Clear to all of you? Clear to all of you, yes or no? See here how it is drawn, like continuous, like block, it is drawn, which I will not be able to draw. So, I will just show the dots, okay, I will just show the dots. Just remember that this entire thing will be like this, okay, like this, full division is happening, continuous globular embryo we formed, okay, something like this, okay, like this, okay, like this full division has happened, got it. Now, here also heart shape like this, like this, okay, complete division has happened. Now, since I will not be able to draw that, okay, I will just draw this much only. I will only draw this much, okay. Now, most important thing is to know what these are, okay. So, this is your zygote, let us say, and then it is going to form first what, students? Pro-embryo. It is going to form pro-embryo. From pro-embryo, it is going to form your globular, okay, globular embryo. From globular, the division is going to become to heart shaped, okay, heart shaped. Now remember this pro-embryo, between this pro-embryo also you are going to have a 4-cell stage, octate cell, all that is going to be there, okay. We are not going to go into that much. So here we have heart shape, and finally we have your mature embryo. Where did this E come from? Okay, mature embryo. This mature embryo will have few parts. What are they? It will have plumule, which will ultimately form what, students? The shoot. Okay, it will have radicle, which will ultimately form the root, and then it will have cotyledon. Again it is for storage, right? It is where the entire energy source is going to be there. So this is what we just understood from the paragraph. So every paragraph I am trying to explain to all of you with the help of a diagram. Done? This is clear? Very nice. I am going to check your attendance. Wait. Oh, who are the conjus who hasn't marked your attendance? Mark your attendance, jaldi. Faster, faster. Fast, 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 fast. Okay, chalo. Let's do the seed. Okay, this is understood, students. This paragraph is clear. Now, a typical dicotyledonous embryo consists of an embryonal axis. Okay, this is the embryonal axis where it has 2 cotyledons. Can you see 2 cotyledons? Okay, 2 cotyledons. So when I am reading, you look at the diagram only. Okay, the portion of the embryonal axis above the level of cotyledon is going to be called. Okay, above the level of cotyledon is going to be called the epicotyle. What is it going to be called? Epicotyle. So if I basically, if I draw something like this and something like this and like this, the portion above is going to be epi, the portion below is going to be hypo, correct? Above is epi, below is hypo, correct? That is all they are trying to explain here. That is, the embryonal axis above the level of the cotyledon is epicotyle, which terminates in the plumule. So where is the plumule, students? See, it terminates in the plumule, okay, or the stem tip. The cylindrical portion below the level of cotyledon is hypocotyle, which will end in what? Which will end in your radicle, okay. Now the root tip is covered with a root cap. Why it should be covered? Because it is going through very difficult life just like us. It has to go through hard rocks, it has to go through soil which is hard, which is not moisturized. So to go through that, to not damage the tip, because you know in the apical regions meristems are going to be there, which are actively dividing cells. If those cells get damaged, then there is chances that the plant will stop growing, right. So to avoid that, we have to give a root cap for this root, root cap. So the root cap is here, clear? This is the structure. This is what is happening. Now this is just the embryo, okay. We are only talking about embryo, we haven't reached the seed, okay. This is the colorful picture which is looking white for all of you, I believe. So plumule, cotyledon, hypocotyle, radicle and root cap. Here if you see, this is your scutellum, coleoptile, shoot apex, epiblast, which is actually a region which has your perisperm, okay. So epiblast, I think one NEET year they have asked this question, if I am not wrong. I do not know which NEET year exactly, but yeah, one of the NEET years these people have asked this question, okay. I don't know how many of you remember that. I think 2020, yeah, 2020 I guess. Yes, ma'am. Okay, understood? Okay, now epiblast, radicle, root cap, coleorhiza. Now students remember, coleorhiza is going to be like a protective region for your radicle, that is your root, okay. And here what is it going to be? Coleoptile. Tile is going to be the one that is going to protect the shoot apex. So root apex, coleorhiza, shoot apex, coleoptile, tile, okay, tile. Now, okay, see, look here, embryos of the monocotyledons possesses only one cotyledon. We know why it's called monocot, one cotyledon, okay. In the grass family, the cotyledon is called scutellum. So, the cotyledon that you see, okay, the region that cotyledon that you see is called the scutellum. They also say scutellum is a portion that separates the endosperm and the cotyledon separately, but then again it is a scutellum in your monocot that is situated towards one side, which is that one side, lateral side. These kind of things you have to be careful, because they can change as assertion, reason, statement based and give you. Instead of lateral, they can give you ventral, dorsal, and without reading we end up answering those questions. Okay. Lateral side of the embryonal axis. At its lower end, the embryonal axis has a radicle and a root cap, okay, enclosed in an undifferentiated sheath called as the coleorhiza. I told you it is like a protective sheath for the root and the root cap. The portion of the embryonal axis above the level of the attachment of the scutellum is called as epicotyle. The epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure called as the coleotyle. Okay, shoot apex. What is this job? The apical shoot meristem or your shoot apical meristem SAM has ultimate job of forming the leaf. Okay, leaf, then later it will become the flower meristem, floral meristem, which will form the flowers, right. So, that is all kept properly inside the coleoptile. Okay. Now, next thing about your seed. Okay, about your seed. Now, students, when we drew our anatropous ovule, okay, all of you remember the anatropous ovule? I drew it like this last time, so I will just draw it like this. Okay, look here students, this is your ovule, correct? This was your ovule before, now also this is your ovule, and here we will have your Nucellus cells. Like this, Nucellus cells will be arranged. Okay, correct? Yes or no? Now, I am going to label each part and tell you what happened to these people. Okay, so here these are your integuments. These are your integuments. The portion where integuments is absent, right? This portion where integuments was absent is called the micropyle. Okay, and opposite to the micropyle you have your chalazal end. Okay, this is your nucellus. Correct students? Then funicle, hilum, all of that is there. But what just happened to this? Okay, what happened to this? Now look here. Now look here. What happened to these people, students, when they form the seed? Okay, they form the seed. They form the seed. One outer coat. Okay, one outer coat. One inner coat. Right, this is the seed, and then in middle they have few portion, like a small portion like this. Okay, so now look at this. This is your seed, and students, what happened? These integuments ultimately became the seed coat. This integuments became seed coat. That is what? Testa and tegment. Testa and tegment. That is the seed coat. Clear? Testa and tegment. So, integuments became the seed coat. Now, the micropyle, which is a small portion where the integuments are not there, that is this end. This is your micropyle. Micropyle, students, in the seed, it became a region for entry. It is given clearly in your NCERT, entry of oxygen and water. Okay, it became a region, it became a small opening that allows the entry of oxygen and water. Clear? Yes or no? Epiblast is actually, even in animals we say after gastrulation, there is a small portion that is formed. Okay, that is your epiblast. Clear? Now here, look all of you, this micropyle, okay, entry, oxygen and water. Why, students? Because integuments were absent in that portion. Obviously, here also if they are forming the testa and tegument, that region integuments are not going to be present. So, this region became the micropyle. Okay, this region became the micropyle region where there is an entry of water as well as oxygen. Okay. Now, what happened? Now, students, this nucellus cells, in case it becomes persistent, in case this Nucellus cells become persistent, we call that as a perisperm. In case this Nucellus becomes persistent, that means they do not leave. They are like, it's okay, whatever happens, I am going to stay with you, like their best friend that we did not ask for. So, they are going to stay with us. That kind of a system, here it is going to be perisperm. Perisperm, that means persistent Nucellus, or I will just label it as Nucellus first and then I will write it down, okay. This is your Nucellus, okay, Nucellus. Now, so what did I say you? If the Nucellus is persistent, students, you have to have this persistency when you crack NEET, okay, that whatever happens in life, I am going to continue this journey, okay. If that persistence is there, then it is possible. So, today what happened, I was just scrolling YouTube and I saw a video of one kid, okay, I do not know the name, I do not know if this kid is a medical influencer, nothing I know. So, this kid has done MBBS abroad in Ukraine or something, okay, and then that kid did MBBS abroad, I think, I do not know how many years, it is not mentioned, and then the war started, okay, and almost a year of no contact, no communication, the kid studied, okay. Then after that, the kid came back to India and then again prepared for NEET, okay, again prepared for NEET and cracked NEET and got into one of the GMCs. That is when I realized how much students are dedicated for this. And students, imagine this kid has taken already drop to get into the first NEET and get into MBBS abroad. After that, dropped out from there, maybe I think 2 years she would have completed. After that, the entire NMC changed the rules. Maybe 2 or 3 years also she spent, I am not sure. Then she comes back to India, takes 2 drops. First drop she got 300, then takes 1 more drop, gets into GMC. Just imagine that kind of dedication people have. I looked at the reel for like 5 minutes straight and I was like mind blown. That energy to still, after one big thing that happened, she came back, she again wrote NEET, again she tried and then she got into a government medical college. I am like, what? Okay. I will be teaching zoology. I will be teaching zoology for sure. Okay. Zoology is like botany students. Sometimes when we get bored, no, to come back, it's like that friendship. Okay, to come back. That is what the botany is. Okay. Yeah, I know MBBS is 6 years, but this kid did not complete. This kid stopped in the middle and came back. So, I was just shocked the whole time. I was just watching the shot over and over because many people tell that teachers and coaching industry push for drop culture, but that is when I realized that thing is not at all true, because drop culture is actually pushed by the students, because there was instance where I told one student, don't take drop, okay, the student has already done the third attempt only. First time the student is meeting me and she gets to know about Vedanta, NEET, English and all of that. So, she told me, ma'am, I am going to take a drop. I told, please do not take a drop, think of something, maybe do a partial drop or think of another course. There are a lot of other courses that pays well, that gives you the same respect and everything. She told, ma'am, how can you tell me not to take a drop? And I was like, okay. So, it's the students who put us in a pressure that tell that please tell me to take a drop. It's not the coaching industry, okay. It's not a coaching industry, it is the students, okay. So, that's the thing. That's what I was realized after 3 years of talking to students. I have got mixed response. Okay, some students tell me that, ma'am, yes ma'am, I will take partial drop, then again end up taking drop. No problem in taking drop, but you should have that motivation. 6 months ke baad, they'll tell me, ma'am, it was a wrong decision that I took a drop. So, that is what is the problem, okay. Baswaraj sir is there. Why Rani, you are not able to see here? Baswaraj sir is there, no, in the channel. So, here persistent Nucellus, okay, persistent Nucellus, we call this as perisperm. Perisperm, okay. Example is going to be beet and black pepper. BB house, you remember. Beet and black pepper. They have persistent Nucellus, okay. Because students, Nucellus was supposed to provide the nourishment for the growing embryo sac. After that, it should have vanished, but it did not vanish, just decided to stay back. So, if it decided to stay back, okay, it it was like, okay, I will stay back. So, persistent perisperm, okay, persistent perisperm, yes, beat, okay, beat, very good, double B, yes, BB house you can remember. So, this is clear to all of you what from seed formed this, yes. Now, students, next one is non-albuminous and ex-albuminous or non-albuminous and albuminous. One second, because this, I think this NCRT print is little too bright, okay. Okay. Now, two types of seeds are there, okay, seeds, one type and one more type, okay, yes. So, this is one type of a seed and this is one type of a seed, okay. So, what happens, students, here, this seed, okay, entire seed, okay. I will tell you, first let me draw, okay, first let me draw, by then all of you quickly mark your attendance, okay, by then we will finish the drawing. Okay, look here all of you, okay, look here all of you, can you all see the colors properly? So, here what happens, students, one set of seed, okay, one set of seed, this basically endosperm was there, correct, endosperm was giving everything for the growing embryo. So, in this you will notice that there is no endosperm, right, no endosperm, that means the seed has utilized all the endosperm, okay, the seed utilized utilized entire endosperm, okay, it utilized entire endosperm. So, this kind of a seed what will you call? What will you call? Quickly tell me in the chat box, quickly tell me in the chat box, what will you call this seed? Okay, this seed we call it as, anyone in the chats? Who is this? Yeah, any Telugu girl? One second. One second, students. Okay. Good job, good job. I can see some answers. I can see some answers. Yes, very good. Non-albuminous, right? Non-albuminous seed, that is, these people do not have any endosperm left. They are like, I used up all the endosperm that was given to me and I utilized all of it, okay, I utilized all of it. So, this is going to be non-albuminous. So, here students you can remember PUBG, okay, PUBG, P-U-B-G, okay, U I am writing small, P for P, okay, B for beans and G for groundnut. Okay, here one exception is there, exception is going to be castor, okay, castor, because this is dicot, but still it's going to be a exception, castor, okay, done, yes, good job, okay, all of you drink some water quickly. Done. P bean, groundnut, castor, okay. You can remember PUBG students, it's easy, it's easy. Yes. Castor, is albuminous. No, here orchid would have come, exception. So sorry. Castor is a dicot, but it comes under albuminous, right? So that is non-albuminous. So here we will write castor, okay? Castor is albuminous. Castor is albuminous. No, castor is albuminous. What is given in your NCERT? You have to see once. Okay. Not complete. See, albuminous seed retain a part of the endosperm. It's not completely used. Example, castor. Okay, castor, even though it is a dicot, it is albuminous. Okay, that is why it is exceptional. I should have written it that side, but by mistake I wrote this side. Okay, like that you have orchid also as an example. Okay, we will see here if it is mentioned here. Okay, now come back here. So students, here what did I say you? I told you that here they have no endosperm left, no food for the later, nothing. Okay, food for later, nothing. Here students, endosperm retains. Okay, endosperm retains. Endosperm retains, that means they keep the food for the later. Okay, seed does not utilize entire endosperm. Okay, it does not utilize entire endosperm. This is the endosperm. Clear? So here what will be the example? So this is called as albuminous. Albuminous. Okay, clear? Albuminous seeds. Or you can also call endospermic and non-endospermic also, no problem in that. Okay, endospermic, non-endospermic, ex-albuminous, non-albuminous, all this many words are there. Okay, yes, clear? Yes. Now, who is that? Yeah. So here albuminous seeds. Here I think you can remember cafe coffee day, cafe coffee day, castor, coconut. I think cafe coffee, only CC you can remember, I guess. Okay, cafe coffee day. Remember cafe coffee day trick I had made for all of you. So many examples are there. Okay, wheat, maize, rice, castor. But yeah, anything you can remember. Barley is there. Okay, ABCD is there. Done? Is this clear? So two types of seed. Okay. Now we will go back to NCERT and look through NCERT as well. So in angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Correct, no students? Fertilized ovule is what a seed is. Now seeds are formed inside the fruit. The seed typically consists of a seed coat, cotyledons and an embryonal axis. The cotyledons of the embryo are simple structures, generally thick and swollen due to the storage of food. So you notice at every stage there is a storage of food, right? At every stage there is a storage of food. Clear? Now, as in legumes, mature seeds may be non-albuminous or ex-albuminous. That means non-albuminous seeds have no residual endosperm as it is completely consumed during the embryo development. Example is going to be pea, groundnut. So PUBG you remember. Albuminous seeds retain a part of the endosperm as it is not completely used up during the embryo development. Example is going to be wheat, maize, barley and castor. Occasionally in some seeds such as black pepper and beet, there is going to be persistent Nucellus, which is your perisperm. Clear to all of you? This entire thing I have explained to all of you. So I hope this is clear. Okay. Persistent, example done. Okay. Now here students, look here. Integuments of ovules harden as tough protective seed coats. The micropyle remains as a small pore. This I told you, no, integuments of the ovule. So this paragraph, this is the diagram. Okay, I will just drag it down here. So when you are reading, you will understand. Okay, yes, here. Okay. So integuments of the ovules harden as tough protective seed coat, just like this. okay, like this. I told you, no, this integuments, they are going to harden and they are going to form the seed coat. What are they going to form? Testa and tegmen, that is going to be the seed coat. Okay. Now they are telling the micropyle, because see students, in the micropyle, in the starting only we notice that integuments are not there, integuments are absent. So if there is a region like that, if it has to become a seed, it is understood that integuments are not going to be present. It's going to be a small opening where there is going to be oxygen and water exchange. Okay. So that micropyle again is going to remain here as an opening. Okay. That is going to remain here as an opening. So the micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed when during germination. Okay, during germination. As the seed matures, its water content is reduced and seed becomes relatively dry. Okay, seed will become dry. How students? 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactive dormancy. That is, if the condition is not favorable, the seed will go for a complete sleep. Now, sleep does not mean technically, it means it will dry out all the moisture in it, because moisture will lead to, harming the seed. So, it will dry out all the water content and it will go to a complete state of rest. Or if favorable conditions are available, that means adequate moisture, oxygen, everything is there, it will start germination. In case that did not happen, let us say the seed went and fell on a rock. It knows that favorable condition is not going to come until it goes and lands on a soil. So, it will wait for favorable condition. So, during that wait, it will make sure to remove entire water content from the seed. Okay? Yes, a stage of rest. It will remove the entire water content from the seed and it will go to a dormancy stage. Like how we have in frog hibernation, estivation, like that. One summer sleep, winter sleep, no? Like that the seed is going to complete, pachak stage. Okay, like this full stick it will become, removing the water content, and when the condition becomes favorable, it will start germinating. Okay, suitable condition, they germinate. Okay, favorable condition comes, they will germinate. As ovules mature into seed, the ovary develops into fruit. That is, the transformation of ovules into seed and ovary into fruit produces simultaneously. Okay, precise, that means simultaneously both are going to happen. The wall of the ovary develop into the wall of the fruit, called as the pericarp. Endocarp, mesocarp. Okay, so like that. The wall of the ovary forms the pericarp. Okay, so we have the fruits may be fleshy, as in guava, orange, mango, etc. Or in most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. Okay, they will degenerate and fall off. However, in few species such as apple, strawberry, cashew, the thalamus also contributes. Here students, we are going to talk about true fruits and false fruit. Wait for a minute. Okay. Okay. Here what is going to happen students? Few things that you have to write. Okay, few things that you have to write. So, here remember ovule, fertilized ovule is called your seed, correct? Okay. Now, ovary to fruit, okay, ovary to fruit and ovule to seed. Okay, this both is going to happen simultaneously. Okay, this is important. Both will happen simultaneously. Okay, it's not like one after the other, one will wait and another person will wait. No, ovary to fruit and ovule to seed will happen simultaneously. Now, students, these are the few things that students usually forget or tend to forget in the last. Integuments are going to form the seed coat. Okay, integuments are going to form the seed coat, and here what is going to happen? Wall of the ovary is going to form the fruit wall. Okay, that is called what? Pericarp. Very, very important. Pericarp. Okay, pericarp. Done? Drupe fruit. We will come to that. Okay, we will come to that. Can you remember these points? Is it easy to remember? Fertilized egg, ovary, ovule, integuments, okay, wall of the ovary. Okay, this much you remember. These are very, very important. Okay, pericarp again. False fruit, true fruits we will come into. We have students have taken the scientist also, so we will go through that as well, so that we do not have, we shouldn't have missed anything by now. Okay, now here two types of fruits are there, very simple. Okay, if you want, we will just divide the page into half and study that. We do not even need one whole page. Okay, it's very small thing. Okay, two types of fruits are there. Okay, two types of fruits are there. What are they, students? True fruits. Now, what do I mean by true fruits? I am expecting this fruit to form from the ovary, and then you have false fruit. So, when I say true fruit, what am I expecting? Ovary should become the fruit. So, if ovary is becoming the fruit, right, if ovary is becoming the fruit, I will call it the true fruit. But if other than the ovary, thalamus, receptacle, any other portion is becoming the fruit, I will call it the false fruit. Clear to all of you? So, in few, okay, in few, there are like fleshy fruits are there, again dry fruits are there, that is separate, students. But here what am I talking about? Fleshy fruit and dry fruit you will be studying in morphology, so don't worry. Now look here, students, when you talk about true fruit and false fruit, if ovary becomes the fruit, I will call it true fruit. If let us say any other part other than the ovary become the fruit, I will call it the false fruit. Now you are true NEET aspirants. Every day you study. There are a lot of false NEET, fake NEET aspirants. That is, they make someone else write their paper. They try to, scam. All that people are called as fake people, right? So here true fruits means, okay, true fruits where the ovary forms the fruit. Okay, ovary forms the fruit. False fruit means other parts excluding ovary forms the fruit, like thalamus in apple. Okay, apple. Clear? That is this clear? Okay. So here false fruit and true fruit is clear to all of you, I believe. So however, in few species such as apple, strawberry, cashew, the thalamus also contributes to the fruit formation. So such fruits are called as false fruits. Okay. Most fruits however develop only from the ovary and they are called as true fruits. Okay. True fruits are the ones that come from the ovary. Okay. Now although in most of these species fruits are a result of fertilization, there are few species where without fertilization the fruit formation will happen. Okay. Without fertilization the fruit formation will happen. So here students, just remember there is also fleshy fruits. Okay, and dry fruits. So here we talk about how the mesocarp, endocarp, pericarp, all this formation is there, and in different fruit like mango, consumption of these places are going to be different, right? So in some, okay, mesocarp, one second. Normally what should have happened, students, in a fruit? Normally, let's say, this is one fruit, okay? It's not an apple, don't consider it as an apple. Okay, this is a fruit. Okay, this is a fruit. Now, what should have happened? We should have consumed the mesocarp, right? We should have consumed the mesocarp. Okay, we should have peeled off the pericarp and the seed here. Inside we would have had a seed. Correct. This is what should have happened. But what happens in some of them? Okay, in some of them we have drupe fruits where each pericarp, mesocarp, endocarp will not be as we expect. Maybe the mesocarp is what we do not, we throw out, endocarp is what we consume. So, like that different fruits are there. Okay, different fruits are there. Now, you only know the drupe fruit. Okay, you can remember drupe fruit, you can remember. Again, more about it, this you will study in your morphology of flowering plants. Okay, morphology of flowering plants. Again, you can remember mango, you can remember coconut, okay, etc. Now, dry fruits, students, you know there is no water content at all in this dry fruits. The entire water content will be dried out. Okay, complete water content will be dried out. Okay, clear to all of you? Okay, perfect. So, students, just give me 5 minutes. Okay, I will just take my medications and come back. Okay, you just resume back in 5 minutes, max 10 minutes. Hello students, let's continue. Okay, this is done. 1 second. Yeah. Now, fruits that are formed without actual fertilization. Okay. So, fruits develop without fertilization. We just told, no, that a fertilized, that is a fertilized ovule, ovary, everything we spoke about, that fertilization is necessary for them to become the seed or the fruit. But there are few fruits without actual fertilization, they do undergo, that means they become the fruit, like in banana. So, here such fruits are called as parthenocarpic fruit. Okay, such fruits are called as parthenocarpic fruits. Example, banana is one such example of a parthenocarpic fruit. Now, parthenocarpy can be induced through application of growth hormones. That means, if like lot of people do it nowadays, students. See, every farming technique is not authentic. Everything that you see outside is not authentic. Half of them are parthenocarpic fruits. Okay, that is why you notice that the fruits, once you cut the fruit, you notice that the fruit is completely pale. They will not have seeds. All this is because these people are inducing growth hormones into them so that the ripening becomes faster. Okay, ripening, and also we get seedless fruits. Okay, seedless fruit. People have a problem with watermelon having seeds, no, and people have also invented watermelon without seeds as well. But it does not have the same flavor and value just like the ones which are formed naturally. So, this is your parthenocarpic fruit. Now, the next most important technique or a word used is apomixes. That is, although seeds in general are products of fertilization, a few flowering plants such as some species of Asteraceae and grasses, okay, and grasses have evolved a special mechanism to produce seeds without fertilization, which is called as apomixes. What is a fruit production without fertilization called as? Thus, apomixes is a form, so fruit without fertilization, parthenocarpy. Apomixes is a form of asexual reproduction, that is, students, where they are mimicking. Okay, they are mimicking sexual reproduction. So, this can mainly happen in two stages. One is where the embryo does not undergo meiosis and directly the formation happens. Second thing is where the nucellus is diploid, that directly becomes the embryo. Understood? Two scenarios where the megaspore mother cell and entire MMC undergoing meiosis will not happen, directly embryo will be formed, or the nucellus cell will become the embryo. This kind of situations can happen. So, this again I will teach you in polyembryony as well. So, there are several ways of development of apomitic seed. In some species, the diploid egg cell, okay, see, this is what I told you, the diploid egg cell is formed without reduction division, that means without meiosis, and develops into embryo without fertilization. More often, as in citrus and mango, okay, as in citrus and mango varieties, some of the nucellar cells, okay, surrounding the embryo sac start to divide and protrude inside to become the embryo. Okay, so they protrude inside into the embryo sac and develop into the embryos. Clear to all of you? Yes or no? Two scenarios: diploid egg cell does not undergo a meiosis and directly form the embryo, or the nucellar cells which are around the embryo sac protrude inside, divide and form the embryo. Clear? Now, in such species, each ovule contains many embryos, right? Many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony, which we mainly see in orange, citrus fruits. Many embryonies, okay, polyembryony. So, take out some seeds of the orange, squeeze them, observe many embryo, different shapes and size, you will be able to observe. So, students, you have studied two things. What is it? Apomitic seed, apomixes and polyembryony. Apomixes, where the seed formation happens without fertilization. Fruit, seed, difference. Fruit, parthenocarpy, seed, apomixes. Polyembryony, where more embryos, more than one embryo is present. Clear to all of you? Yes or no? Give me a thumbs up quickly in the chat box. Quickly in the chat box, give me a thumbs up. Done? Okay. Hybrid varieties of several, so we are coming to an end of the chapter. Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendous increased productivity. One of the problems of hybrids is that hybrid seeds have to be produced every year. If the seed collected from hybrids are sown, the plants in the progeny will segregate and do not maintain the hybrid characters. Production of hybrid seeds is costly, hence the cost of the hybrid seeds becomes too expensive for the farmers. If these hybrids are made into apomix, okay, that means seed without actual fertilization, there is no segregation of characters in the hybrid progeny, then the farmers can keep on using hybrid seeds to raise new crops. So, like when a hybrid is formed, they notice that the progenies are not catching up the hybrid character. But if you do apomixes, you use apomitic seeds, then you notice that the progenies are continuing to have the hybrid characters. So, then why don't we use that to save money, and also the farmers every year can continue the same cultivation, is what they are trying to say. Because of the importance of apomixes in hybrid seed industry, active research is going on in many laboratories around the world. Understand the genetics of apomixes and transfer the apomitic genes into the hybrid variety. So, if we directly do like gene therapy, no, directly we manipulate the gene, like how we do in transgenic animals, biotechnology, recombinant DNA technology. If we do the same apomitic genes directly into the hybrid variety, then we do not even have to think about the seeds. Okay, that is what they are trying to say here. Now, students, just overview of the scientist as well. I think the brightness is too high. I do not know how to reduce it. Students, if I reduce, I think I will not be seen. Okay, so there are multiple problems. Now you see, I don't think I will be seen, but the words are seen, no? Yeah, I look super dark. That's the problem. Okay, so here, see, born in November, Panchan Maheshwari. Okay, Panchan Maheshwari, born in November 1904 in Jaipur. Rose to become one of the most distinguished botanists not only in India but the entire world. He moved to Allahabad for higher education, where he obtained DS, that is basically Doctor of Science. It's considered sometimes above PhD in some places. Okay, during his college days, he was inspired by Duggan, an American missionary teacher, to develop interest in botany and especially morphology. His teacher once expressed that if a student progresses ahead of him, it will give him a great satisfaction. These words encouraged Maheshwari to inquire what he would do in return for the teacher. Okay. Now, he worked on embryological aspect, popularized the use of embryological characters in taxonomy. So, I have just underlined what was his work. Okay. What did he work on? Embryological aspect and popularized the use of embryological characters in taxonomy. He established the Department of Botany, University of Delhi, as an important center for research of embryology and tissue culture. He also emphasized the need for initiation of work on artificial culture of immature embryos. These days, tissue culture has become a landmark in size. His work on test tube fertilization and intra-ovarian pollination won worldwide claims. Okay. Now, he was honored with fellowship. So, these things you have to remember. Okay, they can ask you which was the scientist who got a fellowship of Royal Society of London, FRS. So, that was for him. Okay, Indian National Science Academy and several other institute of excellence. Not only that, he also got Indian National Science Academy recognition. He encouraged general education and made a significant contribution in school education by his leadership in bringing out the very first textbook of biology for higher secondary school, published by NCERT in 1964. Just a read, students, nothing important. Okay. Now, nothing what is that important he has told, or like any definition, nothing is given, only they have just told about his work. Okay, only they have told about his work. I hope it is clear. Yes, it is not very important, but it is good to know who is the person who got that excellence award and what happened. Okay, what happened, that is important. Clear? Done? Chapter is over. Chapter is over. So, students, this pre-fertilization, post-fertilization, everything is done. I hope you will all go to the channel and watch as well. Okay, so see here, go to the channel. And why aren't you people asking, ma'am, what happened to the logo? Where is your photos? What is this Val celebration? So, students, I want to see you all on Saturday 21st at sharp 6 p.m. on our channel, because we are going to felicitate and like we are just going to thank all the parents, all the students who made Vedantu proud in both NEET and JEE category, Olympiad category. They won gold medal for the country. So we are going to felicitate all the students, and I want all my 2026 aspirants to be there, because students, when you see someone, you know, achieving, talking about their success, toppers telling you their strategies, you should be there, because that motivates you to become a better person tomorrow. So I want all of you to think that yes, tomorrow or next year I want to be with Gopika Ma'am, Basavasa, Capto and Diksha Ma'am and celebrate that moment. So that is what I aim to. So when we are talking to those toppers, I want you to see yourself there. So at sharp 6 p.m. on Saturday, I want all of you to be here for this session here. Before that, go and smash the like button, okay? So I want to, we have to show the entire Vedantu how proud our NEET students are for their seniors, okay? Now students, if you go here, part, this is the last part, okay? This is your part 2 and this is your part 1, okay? So part 1, part 2 and part 3, clear? So 3 parts we have finished this chapter. Next I will be doing Animal Kingdom. I want all of you to go and mark your attendance here as well, okay? Go and mark your attendance here as well, so that I know that all of you are ready for Animal Kingdom. Very important chapter, 5 marks chapter, okay? Now some of you have been asking me about DNA. So DNA students, here you can see, here why is it missing? NEET pro batches are there, that is 1 year pro batches. Those who are looking for like same offline like you know, feeling in online, then this is the batch for you, pro batch. Here students, you have right now offers running. So here you can see offline like discipline, because here if you move your head, proctor will send message to your parents. First it will give warning to you, after that it will send message, like how we wrote COVID exams. Remember COVID time we wrote all you know, all of our exams in proctor mode. That means if we move from the screen, it used to all like move our eyes this way. They used to say looking away from the screen. So that kind of system we have here in our you know, online thing. So yes, if you want a 1 year pro course, many students have taken it actually, okay? So yes, and 2 year program links are also here, okay? 2 year program link. Now if you are a student who is looking for a very affordable batch, that also we have. Students, whatever you ask for, we have it, but it is your responsibility to actually take it up, okay? So DNA 2.0 has started, where you will be able to get into DNA batch, okay? So you are going to get 40 plus test, full test, Atwa books, instant class doubt solving, okay? All of this you are going to get. View course plan. These are the prices. Prices will be a little fluctuating sometime, but yeah, these are the options that you are going to get, okay? And most important, you have pedagogy, where you will be able to solve questions at your own pace, at your own free time. You can customize your test. Now today you finished one chapter with me, sexual reproduction and flowering plants. You finished human reproduction with Basava sir. Both of these chapters you can put customize that test and write the test, okay? So that also you can do, clear? So all these things you can do. So I want all of you to be motivated to take some decision, okay? I have made a video on partial drop and full drop. You can go check out that video as well, okay? If you are a student who is thinking of should I do partial drop, full drop, then no need to watch any more videos, just watch this one video, okay? Okay, just watch this one video. And here, if you have scored somewhere between 150 to 300 in your NEET 2025, then watch this before taking a drop. And students, partial drop, full drop, I have made a video here. So everything that you need is here. All you have to do is open the channel and see it for yourself, okay? Any doubts? Any doubts? No Sai Nivas, you can just call me ma'am, okay? Yes, any doubts, please ask me in the chat box. If no doubts, which are the notes that you haven't received? Just tell me that. Which are the notes that you haven't received? Come on, quickly in the chats. All good? I want all of you to comment and go. Don't forget, okay? Comment and only go, okay? Many of you are very, very lazy. Very, very lazy. Hindi. Why you people are testing my language skills or what? I have spoken Hindi many times in class, students. Now I don't want to speak, okay? Yes, okay. Thank you so much. Take care. Bye-bye. See you all in next class. Animal Kingdom. Quickly go and smash the like button, okay? And do let me know in the comments how the session was.